Hi, this is Sophia Shaw, and this is the acetylcholine signaling pathway at a neuromuscular junction. First off, acetylcholine is a paracrine signaling, which is a type of cell-to-cell communication using chemical messengers. Acetylcholine is found in motor neurons that are located in the spinal cord or brainstem. To begin signaling, an action potential sends an electrical signal to travel down the motor neuron. This leads calcium to enter the synaptic terminal, releasing acetylcholine. The acetylcholine is then released into the synaptic cleft, which is the space between the neuron and the muscle cell. The acetylcholine is now ready to bind to its receptor. The receptor is a nicotinic receptor. This is a ligand-gated ion channel that is embedded in the cell membrane. Once the ligand binds to it, it changes shape and opens its channel to allow for the flow of ions. Once the acetylcholine has binded to the receptor acts as a channel for sodium and potassium ions to flow. Sodium enters the cell while potassium exits the cell. This flow of ions causes a depolarization of the cell membrane, which creates an action potential that travels across the membrane. The action potential travels to the transverse tubules. The action potential activates voltage-gated calcium channels, which release calcium ions from the sarcoplasmic reticulum. Once the action potential reaches the sarcoplasmic reticulum, the calcium is released. The calcium causes contractile proteins to interact. These proteins, actin and myosin, interact, causing muscle contractions. That is the response from the acetylcholine signaling pathway at a neuromuscular junction.